So hello everyone, welcome back to the session. So in the last class we have discussed about the endoplasmic reticulum. Okay, so in today's class we are going to discuss about the most important organelle, that is the lysosome. Okay. Whenever we are seeing about the lysosome, we always know that this ribosome is called as a suicidal bag, or because of the digestion process will be taking place in the lysosomes. Okay, but the most important other interesting facts as well as the important functions will be dealing with the dealing in the class regarding with this lysosome. Okay, so in the previous class we have we have started in the cell biology chapter. So far we have completed the cell theory, plasma membrane, nucleus, mitochondria, Golgi complex and endoplasmic reticulum. So in today's class we are going to discuss about this lysosomes. Okay, so see here, so lysosomes, so also we have discussed about the endomembrane system. Okay, so endomembrane system, so in the last class we have discussed about that, right? So we have discussed about what is the definition of this endomembrane system and what are all the organelles which are coming under the endomembrane system, and we have studied that mitochondria, ribosomes, all these are not coming under the endomembrane system. Okay, see, just to see here, and this lysosomes is also not coming under the lysosomes is coming under the endomembrane system. Okay, see, lysosomes are membrane bound. See, you should know that this will be the membrane bound. Okay, see, just make a list. Okay, so what are all the list of organelles and what are all the endomembrane system, that is the first thing. And other important thing, whether it will be the membrane bound or not. If it is a membrane bound, whether it is made up of a whether it has whether that membrane is as the single membrane or it is made up of a double membrane. So that is very important. Okay, so lysosomes are membrane bound. Okay, that enzyme rich organelles involved in intracellular digestion. Okay, so lysosomes are membrane bound. Okay, so it is membrane bound enzyme rich organelle which is involved in intracellular digestion process. So you should note three important point in this definition. One is membrane bound, it is surrounded by a membrane, right? Now, and it has, this organelle has enzyme rich organelle. This is very important. Okay, this lysosome have many important enzymes. We are going to see about that, and there are a lot of enzymes. More enzymes will be present in the lysosome. This is the second thing. And why this lysosome has that many number of enzymes? Because so all these are involving in intracellular digestion process, which means all the waste products, all the unwanted materials has been digested within the lysosomes, and all these digestion is all because of this presence of this enzymes. Okay, so that is why it is a membrane bound enzyme rich organelle, and it is involved in the intracellular digestion, right? Now, see, another important thing, so we have studied this lysosome as membrane bound, isn't it? Now you should know that this lysosomes are single membrane vesicles. The question can be asked. Okay, so the question can be asked like this: whether this what are all the single membrane bound organelle, what are all the double membrane bound organelle? You have to make a list of it. Okay, so the question demands from which of the following is made up of a single membrane organelle. So they have given the 4 options, and 3 options will be double membrane and 1 will be the single. This is how the question will be. Okay, and you should know this lysosomes will be single membrane bound vesicle, right? Now it contains the acid hydrolysis, which means what? Acid hydrolysis, which means all the pH, all the enzymes required the acidic pH. Okay, what does mean by pH? Power of hydrogen. See here, this is 0 and this is 7 and this is 14, which means this is 1. Okay, 1 to 7 is considered as an acid, 7 to 14 is considered as a base, and 7 is considered as a neutral. And the enzymes which is present in the lysosomes are acid hydrolysis, which means it can, the pH will be below 7 or 6. Okay, that will be the pH of the lysosomal enzymes. Okay, now what is the enzymes which is going to degrade the proteins, degrading the nucleic acids, lipids, carbohydrates and cellular debris. Okay, those enzymes which are degrading the proteins is called as a proteases, and those enzymes which are degrading the nucleic acid is nucleases, and lipids is lipases, carbohydrates are amylases. Okay, So all these enzymes are present in the lysosomes. Okay, now who discover? Always we start the organelle with the discoverer. Okay, see here, this lysosomes is discovered by Christian D. Dube. Okay, so this is the name of the scientist who discovered the lysosomes in 1955. Okay, now Dube is also discovered the peroxisome. So this is very important. So though which scientist which discovered the lysosome, he is the one discovered the peroxisome also. Okay, now this lysosome is called the suicidal bags of the cell. Okay, see, this lysosomes is called as the suicidal bags of the cell, and it term used by the D. Okay, so this is the term which was used by D. See, in mitochondria we have studied before the name mitochondria. One scientist have named the mitochondria as bioplast. Okay, like in the same way, this D. originally called this lysosome as suicidal bags of the cell, because as it is involving in the intracellular digestion process because of the presence of acid hydrolysis enzyme. Okay, now in exam, how can we expect the question based on the discovery? Okay, see here, question often asks who discovered the lysosome or who coined the term suicidal bags. We can expect a question like this. Okay, now see the occurrence and distribution. It present in all eukaryotic cells. The lysosome is present in all eukaryotic cells. This is the important, because all the membrane bound organelle which is present only in the eukaryotic cells, not in the prokaryotic cells. We have discussed in the basic difference between the prokaryotic and eukaryotic in the day 2 of our class. Okay, see here, this lysosome is abundant in phagocytic cells. Phagocytic cells, we know that the cells which are involving in engulfing the pathogen or microorganisms, and it results in the digestion. That process is called as a phagocytosis process. And the cells which are involving in the phagocytosis process is called as a phagocytic cells. And what are all the phagocytic cells which is present in the lysosome is nothing but the macrophages and the neutrophils, and again which is a type of WBC, right? Okay, and hepatocytes means what? Liver cells. Okay, the cell which is present in the liver is called as a hepatic cell. So always whenever we are heard about the word hepato, you have to relate that term with the liver. Okay, now the kidney cell. Okay, the cells which is present in the kidney, right? So this lysosomes are abundant in phagocytic cells like macrophages, neutrophils, hepatocytes, kidney cells. Why the liver cells, kidney cells and macrophages and neutrophils have more number of lysosomes? Because these are all the cells are in need of the lysosome to see. In case of hepatocytes, it has to digest the toxic materials. Okay, liver cells, detoxification will be done in the liver. So obviously lysosome are in need in liver cell. Okay, kidney cells, again the lysosome will be need in the kidney cells, and macrophages and neutrophils, they are involving in the phagocytosis process. So the lysosomes are in need to lyse the microorganisms. Okay, so why all these cells are enriched of lysosome means these are all the reason, because it has to involving in lysing the microorganisms. Okay, so this is very important. Now see here, very important, we have studied that nucleus is not present in RBC, mitochondria is not present in RBC. Like in the same way, Golgi complex is not present in RBC, endoplasmic reticulum, see here, all these organelles are not present in RBC, no doubt in that. In addition to all those organelles, this lysosomes is also absent in mature RBC, that is the red blood cells. Okay, and see, plant cells, a large central vacuole performs the similar role like the lysosome in animal cells. So that is why the lysosome is not present in the plant cells. Okay, why? Because vacuoles is also doing the same function. Then no point of having the lysosome in the plant cells. This is the reason. Okay, so in plants, the lytic vacuole, functional equivalent of the lysosome. Okay, let me show you the picture of this lysosome. I forgot to add the picture, one second. Just a minute, let me show you. Let me show you the clear picture of it. See, this is what the let me copy into this 1 minute. I'm sorry for this. It's okay. Let me show you in tomorrow's class itself then. It's okay. Okay, see here in plants the lytic vacuole, that is functional equivalent to the lysosomes. Okay, so what they are trying to say is, in plants what are all the function done by the vacuole that will be done by the lysosome in case of animal cells. That is why lysosome is not present in the plant cells. This can be expected in the question. Okay, now comes to the ultrastructure of the lysosome. Okay, see here the size and shape, what will be the size and shape of the lysosome. See here it will be 0.2 to 0.5 micrometer. Okay, so this is what the size of the lysosome. And what will be the structure? Again it will be the oval or the spherical shape. This is what the structure of the lysosome. The electron dense matrix under the TEM. Okay, so TEM is the type of electron microscope, transmission electron microscope. So the electron dense matrix can be, which is present in the lysosome, which is clearly seen under the TEM. Okay, now so again it will be the single unit membrane. Okay, there is only one membrane will be present, single membrane bound organelle is lysosome. Okay, and it is rich in the glycoproteins. Okay, carbohydrate which is associated with the protein, that is what the name glycoprotein. Okay, see this is the protein which is a glycoprotein which protects the membrane from self digestion. So here comes a question. See, lysosomes are involving in the enzyme which is present in the lysosome has a capacity to lyse the subproteins or carbohydrates and whatever be it. Okay, but why the lysosome itself is not affected by the enzyme with its enzyme present within it. Okay, so all because of this glycoproteins which is rich in the lysosome. So this glycoproteins is the one protecting the membrane from the self digestion. So this is very important. Okay, now it contains the proton pumps. So as we have mentioned before, as we have studied about this proton pump in the mitochondria itself, you should know that we have studied the enzyme which is present under the in the lysosome is an acid hydrolysis, which means what? That enzyme prefer only the acidic environment. Now who is responsible for maintaining the acidic environment in the lysosomes? That all because of this VATPases. Okay, vacuolar ATPases. So this VATPase is involving in pumping the proton. Pumping the proton sense means what? Proton means H+. So if the H+ concentration is higher, so we can say that as an acidic environment, right? So this is very important. So as because of the presence of this VATPase transporter, that is which contains a proton pump, so this is the one maintaining the acidic pH in the lysosome. So you should know what will be the exact range of pH of an enzyme which is present in a lysosome. That will be approximately 4.5 to 5. Okay, so this is what the acidic pH. Some enzymes may vary. Okay, so maintained by ATP dependent proton pump. Okay, so ATP dependent proton pump. So this VATPase is involving in pumping the proton. So that is why it maintaining the acidic environment in the lysosome. But to pump the proton, this VATPase pump needs an energy. So that is why in the form of ATP. That is why we are calling them as the ATP dependent proton pump. This is nothing but VATP. Okay, so how in the exam how can they trap us? So pH is acidic, not neutral. Never ever it is neutral, it is acidic environment. Okay, now what will be the chemical composition of the lysosome? So major components which is present in the lysosome will be the enzymes. We know that, no doubt. Majorly the space occupied by the lysosome will be the enzymes. Okay, so that will be approximately 42 different types of enzyme. It is not the number, it is the type. Okay, approximately 40 to 50 types of enzyme will be present in the lysosomes. Okay, and also the membrane proteins are present and transporters are present in the lysosome. Okay, now as we have studied approximately 40 to 50 types of enzymes will be present in the lysosome. What are all the types of enzymes which is present in the lysosome? We can call them as the acid hydrolysis. Okay, acid hydrolysis. Now the first enzyme will be the proteases. Second, nucleases. Proteases means those enzymes which break the protein as protease. Okay, that is what cathepsins. Right. Now nucleases. So enzyme which is destroying the nucleic acid is nucleases, that is DNA. We are having two nucleic acid, one is DNA and RNA. An enzyme which is breaking the DNA is DNase, an enzyme which is breaking the RNA is RNA. So that is why we are categorized them under the nucleases. Okay. Now lipases, these enzymes which are involving in breaking the lipids. Okay, and glycosidases, these enzymes are involving in breaking the carbohydrates, that is a sugar. The word glyco, it refers to sugars. Okay, and phosphatases, and those enzymes which are breaking the phosphate group is phosphatases, and sulfatases, those which are breaking the sulfur group. This is the sulfatases. Okay, see, the main important key point here will be the all enzymes are acid dependent. So this is very important. Okay, now biogenesis of lysosome. Whenever we are studying about the Golgi complex, we have studied one statement. This Golgi complex is formed from the formation of Golgi complex is due to the some pieces will be break from the rough endoplasmic reticulum, that results in the formation of vacuoles and the vesicles. And the vesicles altogether we are calling them as the rough endoplasmic reticulum. And this endoplasmic reticulum, which means the formation of Golgi complex. From the rough endoplasmic reticulum, Golgi complex will be formed, and from the Golgi vesicles, this is the one involving in formation of the lysosomes. Okay, so for the formation of lysosome, Golgi complex is very important. Okay, see here, the enzyme synthesized in rough endoplasmic reticulum. Okay, because you should know that enzymes are what? Enzymes are proteins. Okay, and the protein folding and protein modification will be done in the rough endoplasmic reticulum. Okay, now you should know the enzyme which is necessary for the lysosome is also present in, also formed by the rough endoplasmic reticulum. Okay, you should know that. And it is processing in the Golgi complex, the protein processing. Okay, that will be taking place in the Golgi complex. Only after the processing of the protein will become active, and it becomes the active enzymes when it enters into the lysosome. This is what the scenario. Okay, now it packaged into vesicles and it will be forming the lysosome. Okay, so this is very important. Okay, now very important thing will be mannose 6-phosphate will be the signal. See, the mannose is a sugar. Okay, at its sixth carbon position, phosphate group is adding. So that is what a mannose 6-phosphate. Okay, so the mannose 6-phosphate, whenever a protein is added by the mannose 6-phosphate, then you should know that this protein is going to lysosome. this is the signal. See, from the Golgi complex, the protein has to be entered into lysosome. Okay, so if the protein from the Golgi complex is added by the mannose 6-phosphate, you should know that this protein is destined to lysosomal organelle. This is very important. Kindly make a note of it. And what are all the steps? Okay, before the enzyme has to go to the lysosome, what are all the steps to be happen before that? Okay, see, enzyme synthesized in the rough endoplasmic reticulum. No doubt in that. Okay, everybody is accepting the fact that protein will be synthesized, protein will be folding properly in the rough endoplasmic reticulum. That is what enzymes here, you should know enzymes are proteins. Now modified in Golgi, okay, modified in Golgi, which means what? They are adding the mannose 6-phosphate. This is like a tag, okay. If mannose 6-phosphate is added by the Golgi complex to any proteins, you should know that, then the cell will came in to know that this molecule has to send to the lysosome. Okay, so that is what mannose 6-phosphate tag. Okay, that will be done in the Golgi complex, and it's recognized by mannose 6-phosphate receptors which is present in the lysosomes. Okay, vesicles bud off from the lysosome. So through the means of vesicles, the molecule will be packed and it has to enter into the lysosome. It will be received by the receptors which is present in the lysosome, and the vesicles will be bud off from the lysosome. These are all the steps. Okay, now absence of mannose 6-phosphate, enzyme secreted outside, that is what cell diseases. Okay, one cell disease, it leads to lysosomal cell disease if there will be no mannose 6-phosphate tag. Okay, so this is another important thing, right? Now, and we are having two different types of lysosomes. Okay, we are having types of lysosomes, and that is primary lysosome and the secondary lysosome. Primary lysosome will be the newly formed lysosome from the Golgi complex. Okay, and it contains an enzyme, but you should know this enzymes which are inactive in case of primary lysosomes. This is very important. So as the enzymes are proactive, that is inactive, there will be no digestion starting in the primary lysosomes. Okay, and the secondary lysosome. Secondary lysosome means formed by the fusion of primary lysosome with the vesicles. Whenever the vesicle is fused with the primary lysosome, then it results in the formation of secondary vesicles. So vesicles plays a crucial role in forming of rough endoplasmic reticulum, Golgi complex, lysosome and the secondary lysosomes. Okay, so this is crucial. Now, this heterophagic lysosome, this is the type. There are different types of secondary lysosome, and the first type will be the heterophagic lysosome. Okay, fusion with the phagosome. Whenever the secondary lysosome is fused with the phagosome, we are calling them as a heterophagic lysosome. Digest the external material, example macrophages. You should know we are having different types of secondary lysosome, and the first type will be the heterophagic lysosome. And this heterophagic lysosome is formed whenever the lysosome is fused with the phagosome. Okay, and this heterophagic lysosome are involved in digesting the external material. Okay, now the best example for the heterophagic lysosome will be the macrophages, which this macrophages is the one involving in the phagocytosis process. Now the second thing will be the autophagic lysosome. Autophagic lysosome is also called as the autolysosome. Okay, see, whenever the lysosome is fused with the autophagosome, we can call them as the autophagic lysosome. Lysosome plus autophagosome together, lysosome plus autophagy, we are calling them as the autophagosome. Okay, now this is the one digesting the old organelles. Okay, so the autophagic lysosome, whenever we are undergoing fasting, okay, at that time the old organelles and the waste, old particles, old things, everything will be digested through the process of this autophagy. That is why fasting is very important. In different religion, different modes of reason is there, but the only reason or the scientific fact to induce autophagy, we have to be starved. Okay, so that's what, and if on special occasion we tend to fasting. Okay, Now this is called as an autophagy, which means phagocytosis of our own cells. That is what autophagy. So during starvation, the autophagy will be increased, right? Now, residual bodies. Residual bodies, they contain undigested materials. Okay. See, residual bodies means they are containing the undigested materials. Now, so whenever we are digesting the food, some food has been digested and all the digested food has to be absorbed. Now, some of the foods are undigested and all has to be eliminated from the body. So that is what those undigested material has been present, we are calling them as a residual bodies. Okay, that residual bodies has to be exocytosed. Exocytosed means what? That has to be sent out from the cell. Okay, so that accumulate as lipofuscin granules. Okay, lipofuscin granules. So this lipofuscin, if the lipofuscin will be increased, so this is considered as an aging pigment. So this is very important. Okay, and the residual bodies will be in yellow color. And see here, intracellular digestion. So we have studied the functions of lysosome now. So first one will be the intracellular digestion. So intracellular digestion, they are digesting the cell. Intracellular digestion means they are digesting within the cell. So in the modes of heterophagy and autophagy. Okay, now autolysis means they are destructing our own cells. Okay, our own cells, but that cells are old. Those cells are old, they are undergoing an autolysis process. So they are important. This autolysis process is important when metamorphosis and tissue remodeling. So at these times the autolysis is very important. Okay, now the defense mechanism, they are the one destroying the pathogen. So that is why the lysosomes are also involving in the defense. Okay, it is important in immune cells. Of course it is important in immune cells. Why? Because this lysosomes are involving in phagocytosis process. And another important thing will be acrosome of sperm. It is not a modified lysosome, it is a modified Golgi complex. Okay, so the acrosomes, which is acrosome of the sperm, which is nothing but a modified Golgi complex. It is not a lysosome. Kindly make a note of it. And the development and differentiation. So the cell division, controlled cell division. So we are calling them as an apoptosis. It has an indirect role here. So the development and differentiation means here, this lysosome, whenever the lysosomes are developed proper, the cell death apoptosis process is also proper. Okay. Now see here, the lysosome versus vacuoles. What is the difference between? So as we have mentioned before, the lysosomes are absent in case of plants. Why? Because the vacuoles are present in plants, and the vacuoles is also doing the same function as lysosome in animal cells. Now what will be the difference between the lysosome and vacuoles? So the lysosome is the animal cells. It is present in the animal cell. If it is present in the plant cell, we are calling them as a vacuole, and the size of lysosome will be very small and the size of vacuole will be very large. Okay, and the pH is acidic pH and the vacuole is acidic pH. Okay, and the function of this lysosome is digestion, and the vacuole function will be storage as well as digestion. Okay, and the plant vacuole is also contains the is also containing the acid hydrolases. Okay, so this is important. Now, lysosomal storage disorders. What are all the disorders which is present in the, if the enzyme which does not function properly in the lysosome, it leads to some storage disorders. See here, when the lysosomal storage disorder will appear, whenever the deficiency of specific lysosomal enzymes. So the enzyme which is present in the lysosomes are very important. If the specific lysosomal enzymes are not function properly, it leads to diseases. Okay, storage diseases, and leads to substrate accumulation, because all the substrate which are not in need, that has to be destroyed by the lysosome, and if there is no proper enzyme, specific lysosome enzyme, it leads to accumulation and further it leads to disorder. And see, this is very important thing. the disease which is, what are all the disease related to the lysosome? So this we can expect in the question. Tay-Sachs disease. See, Tay-Sachs disease, which is Tay-Sachs disease is somewhat related to an enzyme, which means related to an lysosome. Why? Because of the absence of an enzyme. See, because of the absence of this hexosaminidase A enzyme, this hexosaminidase enzyme is involving in digesting the GM2 ganglioside. As there will be no point of have no enzyme, this enzyme is deficient, it results in the GM2 ganglioside accumulation. So it results in the Tay-Sachs disease. Okay. Gaucher disease, again it will be the disorder related to the lysosome. This Gaucher's disease is arise due to the accumulation of glucocerebroside. The glucocerebroside has to be digested by this glucocerebrosidase. As this enzyme is not present in the lysosome, there is accumulation of this glucocerebroside more in the lysosome. It results in the Gaucher's disease. Okay. And Pomme's disease, in Pomme's disease, the glycogen is getting accumulated in the lysosome, and it results in the, as there will be no enzyme of, as there will be no acid maltase enzyme. If there will be no acid maltase enzyme, the glycogen will not be digested. It is getting accumulated in the lysosome. It results in the Pomme's disease. And Hurler's disease. Hurler's disease, due to the deficiency of an enzyme alpha urodinidase. Okay, so due to the so as this enzyme is not present, mucopolysaccharide is getting accumulated in the lysosome. Okay, Eissel's disease, as the mannose 6-phosphate tagging is not proper, multiple substrates is getting accumulated in the lysosome, so it results in this disorder. Okay, so this can be expected in the mass of following questions. Okay, which enzyme deficient, what is getting accumulated in the lysosome, and what will be the name of the diseases. Right now, okay, so the regulation and protection mechanisms. Lysosomal enzyme synthesized as inactive precursor. So initially the enzyme which is synthesized in the primary lysosomes, as we have discussed before, it will be inactive, and that will be the precursor, inactive precursor initially. Membrane glycoproteins prevent the self-digestion. So as we have mentioned before, why the enzyme which is present in the lysosome is not digesting the lysosomes first? Why? Because of the presence of the prevention called the glycoprotein. Okay. Now, acidic pH confined the inside lysosome. Okay. Acidic pH confined the inside lysosome. The inner layer of the inner part of the core area of the lysosome is experiencing the acidic environment. Okay. If membrane ruptures, it results in the cell death. Okay. Now, how we can compare the peroxisome with the lysosome? Okay, so the pH of the lysosome will be acidic and the pH of the peroxisome will be neutral, and the enzyme which is present in the lysosome is hydrolysis. The enzyme present in the peroxisome will be oxidases and catalases. This is important, and the function of this lysosome will be the digestion, and the function of peroxisome will be the detoxification. Okay, see, the lysosome will be originated from the Golgi complex, and the peroxisome will be originated from the endoplasmic reticulum. Okay, so this is important. See, in the examination, what are all the very important thing we can often come across with is the lysosome are not double membrane bound layer, it is a single membrane. Okay, and the lysosome acid hydrolysis works at low pH. Yes, of course it is right. Low pH is an acidic environment, 1 to 7. This will be the low pH. And the mannose 6-phosphate is crucial. Of course, mannose 6-phosphate tagging is very important, and acrosome is the some of the resources are saying this as a lysosomal origin, and some will be saying the Golgi complex origin. So you consider this acrosome will be the modification of Golgi complex. Okay, and lipofuscin, this is considered as a residual body. Okay, aging pigment. Okay, now one-liners. So the discovered by this lysosome will be discovered by Christian Didieu. He is the one discovered the peroxisome also. And acidic pH maintained by VTPases because of the proton pump. And mannose 6-phosphate is the Golgi sorting signal that has to be destined to the lysosome. Again, acrosome will be the modified Golgi complex. Lipofuscin indicates aging. Accumulation of this lipofuscin, that is an aging pigment. It indicates aging. So we have completed all the important functions with respect to the lysosome, and the first question will be, lysosomes were first discovered by, and we know who discovered the lysosome. It is none other than the Christian D. Duke. Okay. Now the acid pH of the lysosome is maintained by, we know that B type H plus ATPases is the one maintaining the lysosomes, pH. Which signal is essential for targeting hydrolysis to lysosome? That will be the mannose 6-phosphate signal. Now, failure of the mannose 6-phosphate tagging leads to, see here, if the mannose 6-phosphate tagging is not proper, it leads to eye cells disease. Okay. Now, autophagic lysosomes are primarily involved in, lysosomes are involving in, as the name suggests, auto means self, phagy means phagocytosis process, destruction of the, recycling of old organelles. So this is what an important thing. Residual bodies containing lipofuscin are associated with residual bodies contain lipofuscin. So it results in the cell division, aging, immune response, and cell secretion. What will be the proper answer? That will be the aging process. Okay. Which of the following is a modified lysosome, modified Golgi complex? That will be the acrosome. The next class I will clear whether it will be the modified lysosome or modified acrosome. So as of now, just think it is a modified Golgi complex. Okay, basically it is from both, right? Now, which lysosomal enzyme deficiency causes Tay-Sachs diseases? Tay-Sachs diseases, as there will be no presence of the enzyme hexosaminidase A. It results in the deficiency. Now, in plant cells, the functional equivalent of lysosome is nothing but a central vacuole. Okay. Now, which statement about lysosome is incorrect? They are single membrane bound. Of course, it is right statement. They contain acid hydrolysis. Of course, it is right statement. They function as neutral pH. No, it is in an acidic way. That is a low pH. They originate from the Golgi complex. Of course, it is a right statement. Okay. So that's all about the organelle lysosome. We'll meet you in the tomorrow's class with another organelle. Okay. Thank you so much.